Hi students, in this video we are going to discuss about meiosis or reduction division. Why it is called as reduction division means in this division we can expect a reduction of chromosomal number into half. So that's why it is called reduction division. As well as we call it as a heterotypic division. Heterotypic division means after completion of division we won't expect any similarities between daughter cells as well as mother cell. So that's why it is called a heterotypic division. First time August Weismann in 1887 he observed movement of or number of chromosomes in animals. Means number of chromosomes decreased in animal cells. So he observed that decreasing pattern first time. So Augusta Weismann is the first person he observed which one decreasing of number of chromosomes in animal cells in 1887. Next Strasberger in 1888 in plants he discovered meiosis. So first discovery of meiosis belongs to whom means Strasberger only in 1888. Then after Farmer as well as Moore in 1905 they coined the term meiosis. Means finally we can expect meiosis is discovered by Strasberger and the term meiosis is introduced by whom means Farmer as well as Moore in 1905. Here you need to understand meiocyte. Meiocyte means the cell which involves in meiosis, that cell is termed as here meiocyte. You need to remember here always diploid cells only involves in meiosis. We never expect involvement of haploid cells in meiosis because the number of chromosomes has to be reduced in this division. So we can expect a reduction of number of chromosomes in diploid cells only. We won't expect in haploid cells. More than diploid also we can expect, for example tetraploid. Like that cells also may involve in meiosis. Next types of meiosis. Here we can expect three types of meiosis based on the type of involvement of cell. Here zygotic meiosis, sporic meiosis, another one we can expect here gametic or terminal meiosis. First we are going to discuss about which one means zygotic meiosis. Zygotic meiosis or initial meiosis. Zygotic meiosis or initial meiosis means after fertilization, after formation of zygote, zygote involves in meiosis. Example in thallophytes, haploid life cycle following organisms. After formation of zygote, what happens? They involve in meiosis and they produce which one means haploid cells. These haploid cells they develop as individual organisms. As well as when the fertilization happens again they produce zygote, again this zygote involves in meiosis. Means in these organisms we can expect meiotic division after fertilization. Very important, you need to understand after fertilization we are expecting here meiotic division in zygotic or initial meiosis. Next one is sporic meiosis or intermediate meiosis. In this meiosis type some thallophytes and higher plants also are embryophytic plants. These higher plants are embryophytic plants. These plants involve in which one means sporic meiosis or intermediate meiosis. Last cells of sporophytic generation is considered as here spore mother cells. Before spore mother cells we can expect formation of zygote. This zygote involves in a number of mitotic divisions and produces which one means spore mother cells. These spore mother cells involves in meiosis and produces spores. These spores develops as a gametophyte, means individual organisms. For example, if you consider here Funaria plant, this is a Funaria plant. In this Funaria plant we can expect two phases. This is gametophytic phase, this is a sporophytic phase. This is gametophytic phase, this is a sporophytic phase. In gametophytic phase we can expect a formation of gametes, male and female gametes by mitotic divisions only. We won't expect a meiosis division. After production of male and female gametes, male gamete is termed as here antherozoid, female is termed as egg cell. Here antherozoid and archegonium. Inside the archegonium we can expect presence of which one means female one, that is egg cell. After completion of fertilization we can expect formation of zygote. Before formation of zygote the existing part totally in which condition means haploid condition, means gametophytic phase is in haploid condition. After formation of zygote, that zygote involves in mitotic divisions and produces a diploid life cycle, that is a sporophytic phase. You have to remember sporophytic phase is always diploid in condition. So in that we can expect a foot, seta and capsule. Three structures we can expect. For the formation of these three structures we can expect mitotic divisions. After completion of so many mitotic divisions inside the capsule, we can expect development or differentiation of some cells. That cells are termed as here spore mother cells. These spore mother cells involves in a meiotic division and produces haploid spores. So here inside the capsule we can expect a meiotic division. So you need to understand this is a second type of division, that is sporic or intermediate meiosis. Next, last and final, we are going to discuss about gametophytic gametic meiosis. In this gametic meiosis, we can expect which one means in animals especially we can expect here meiocytes. Namely, we call them as gamete mother cells because they produce gametes. That's why we call them as gamete mother cells. These gamete mother cells involves in meiosis and produce gametes. So that's why this type of which one meiosis is termed as a gametic meiosis or a terminal meiosis. Why we are called these three types of meiotic divisions means first initial meiosis means we can expect lower organisms. Intermediate meiosis means at intermediary level during evolutionary process we observed this intermediate meiosis, and final one which one means now in evolved organisms we are expecting gametic or terminal meiosis. Terminal means at the end of the evolutionary process we expected more developed meiotic division type, that is terminal meiosis. Next, now we are going to discuss about which one means salient features of meiosis. In salient features of meiosis, we can expect first one nucleus divides twice. Means in a division of meiosis, we can expect division of nucleus twice. That's why it produces how many nuclei means 4 nuclei means 4 cells it produces. Chromosome divides only once. Means chromosomes daughter chromosomes formation takes place in S phase. Means a single cycle of DNA replication occurs and it produces duplication of chromosomes. That duplicated chromosomes divides only once in this meiosis. Next, pairing of homologous chromosomes as well as exchange of genetic material takes place between homologous chromosomes. Means you need to understand here pairing of homologous chromosomes happens. This pairing of homologous means one chromosome is derived from a parent, that is a male parent, another one is a female parent. That two chromosomes involves in pairing process. After completion of pairing process they involve in exchange of genetic material as well as we can expect exchange of genetic material between homologous chromosomes only. Next one, 4 haploid daughter cells formed each with half number of chromosomes. After completion of meiosis we can expect formation of 4 daughter cells. They are haploid in condition. So in that we can expect half of the number of chromosomes only present. These are the salient features of meiosis. Regarding stages of meiosis we will discuss in next video, but I want to give a little bit information regarding this meiosis. First if you observe stages of meiosis we can expect 2 stages. One is meiosis 1 and another one is meiosis 2. This meiosis 1 is termed as heterotypic and disjunctional and reduction division. Heterotypic division, disjunctional division as well as reduction division. And meiosis 2 is considered as homotypic and equational division and somatic division. So it is not somatic division, but generally we can expect which one means it is similar to mitosis. Which one meiosis 2 is similar to mitosis. That's why it is considered as this terms, which one equational division, homotypic division. Actually you won't consider as a somatic. Generally when we call it as somatic means generally if it happens in vegetative cells for growth purpose, then only we call it as a somatic. But we may say them as somatic because it is similar to mitotic division. Next, meiosis 1. If observe meiosis 1, here it is a complicated process as well as a lengthy phase. Means it takes more time. Next, karyokinesis 1 and cytokinesis 1 takes place in meiosis 1. Next, karyokinesis 1. What is karyokinesis means? We know that karyokinesis means division of nucleus. First nuclear division in meiocytes happens, then after it follows cytokinesis. Immediately or sometimes we may expect a delay of cytokinesis. Next, in this karyokinesis we can expect 4 subphases: prophase 1, metaphase 1, anaphase 1 and telophase 1. In this 4 phases we can see here prophase 1. It takes place for a longer duration and it is a complex process. So based on the chromosomes behavior it is divided into 5 subphases again which one prophase 1. Means in prophase 1 it is a long time consuming process as well as complex process. So that's why we are expecting how many subphases in this means we are expecting 5 subphases, that is leptotin, zygotin, pachytin, diplotin and diakinesis. Regarding these phases I will discuss later. In that first leptotin and zygotin, these two phases are shorter than pachytin. Pachytin is a complicated phase again in prophase 1. So like these subphases are divided for easy understanding purpose. Remaining topic I will explain clearly from starting to ending point. This is the first part of meiosis 1. Thank you, students.